Regulation of gene expression by vitamin A. Vitamin A serves an important function in nearly every tissue in the body. Its derivatives, all-trans-retinoic acid and 9-cis-retinoic acid, regulate genes. Gene regulation by the active forms of retinoic acid occurs via receptors. Retinoic acid binds to two members of a family of proteins that act as nuclear receptors, RXR and RAR. These nuclear receptors include one special domain that binds retinoic acid and another domain that binds specific sequences in the DNA. Without binding to retinoic acid, these receptors are inactive. Thus, the retinoid receptor works as a transcription factor. Transcription factors are able to bind to DNA and turn on genes. Let's look at the nuclear receptor RAR. RAR alone is not able to bind to DNA. However, through a process called heterodimerization, RXR and RAR come together to form a receptor complex which can bind to DNA. RXR can also partner with several other nuclear receptors, including the receptor for vitamin D, thyroid hormone, bile acids, and fatty acids. Each of these RXR pairs binds to a different sequence in DNA. The retinoid receptors form one part of a larger system of nuclear receptors that are activated by a variety of compounds. This larger system of nuclear receptors regulates a wide range of metabolic signaling to control the expression of multiple genes and affects many tissues. Retinoic acid influences metabolic signaling via DNA regulation. It is able to turn on a large number of genes in a variety of tissues in the body, which accounts for the diversity of actions of vitamin A.